Welcome to Matri Academy. Let's discuss the previous year question paper for ARS NET. हेलो स्टूडेंट्स, अब हम कुछ प्रीवियस ईयर के क्वेश्चंस पेपर ICR नेट और ARS प्री के सॉल्व करने के लिए जा रहे हैं। ये काफ़ी पुराने क्वेश्चंस पेपर हैं 2001 से रिलेटेड और ये मेमोरी बेस्ड हैं। उस समय क्योंकि कोई भी क्वेश्चन पेपर ऐसे अपलोडेड नहीं होता था ऑनलाइन या और कंप्यूटर बेस्ड एग्ज़ाम्स भी नहीं होते थे, बट दीज आर डेफिनेटली आर दोज क्वेश्चंस विच वॉज केम इन यूर प्रीवियस एग्जामिनेशन। सो इसको है इम्पॉर्टेंट पॉइंट्स की तरह मैंने इसको तैयार किया है ताकि आपके एग्ज़ाम में ये हेल्पफुल हो। सो चलिए शुरू करते हैं। हमारा फर्स्ट क्वेश्चन था, इन लिपोसोम मीडिएटेड जीन ट्रांसफ़र मेथड DNA enters the protoplasm due to the process of endocytosis and the correct answer था endocytosis. Arabidopsis, a small unit of ribosco, is a green tissue specific promoter. ये क्वेश्चन बहुत बार आपसे पूछा जाता है। इसका आंसर है ग्रीन टिश्यू स्पेसिफिक प्रमोटर. During agrobacterium infection, the plant cell begins to synthesize an arginine derivative which is known as opines. अब साथ साथ में नोट भी करते जाएं. Enzyme reverse transcriptase is used for making complementary DNA of mRNA. Replication of DNA by rolling circle mechanism give rise to a concatemer. ये भी क्वेश्चन बहुत बार पूछा गया है. Cosmate vector has got the cos site of phage lambda. A relaxed plasmid can have its multiple copies per cell. Restrictions enzyme that cuts straight across the DNA produces the blunt ends. ये सारे मल्टीपल चॉइस क्वेश्चन को मैंने डायरेक्ट आंसर से कोरिलेट करके ये पॉइंट्स शॉर्ट नोट्स में बनाए हैं. इन विट्रो पैकेजिंग इज रिक्वायर्ड फॉर पैक वेक्टर. Lambda DNA has short single-stranded 5' projection of 12 nucleotide length. Ethidium bromide is used for visualizing DNA under UV light. The direct DNA uptake by the plant protoplast is stimulated by PEG, polyethylene glycol. Acetosyringeone is used as an inducer of the Biron operon during agrobacterium mediated gene transfer by co-cultivation method. This method is called as co-cultivation method. Lectin, protease inhibitors and alpha amylase inhibitors all are insecticidal proteins. In agrobacterium mediated gene transfer technique, which antibiotic is used to check the excessive bacterial growth? And the answer is cefotaxime and timentin. The most commonly used technique to develop transgenic plant resistance to viral disease is coat or capsid protein gene. Next, which of the four operons of Viron region are required for virulence functions? And the options are correct options are Viron A, B, D and G. Nucleolus takes part in the formation of RNA. Nuclear envelope breaks down in prophase 1. Plant cells are surrounded by a rigid cell wall made up of polysaccharides. The 9+2 arrangement describes the basic structure of flagellum. Endoplasmic reticulum is a system of membrane-bound channels. Scientists have proposed that small pores must exist in the cell membrane so that water molecules can move freely in and out. Lysosomes are involved in the breakdown of cells and tissue That is why they are called suicidal bags. The transgenic plants of tomato showed delayed fruit ripening due to the suppression of ethylene using the technique of antisense gene technology. The RB and LB flanking tDNA regions are composed of 25 base pair As. Question asked in ARS 2007-2018 also. The gene transferred from E. coli to tobacco to increase the level of mannitol was mannitol dehydrogenase. Method employed to convert the expression of transgene at translational level is Western blotting or Western hybridization. During agrobacterium mediated genetic transformation, the tDNA gets coated by a protein which is the product of Viron E genes. GFP is not an antibiotic resistance gene. It is a scorable marker gene. Endochitinase can be used as an antifungal agent. If adenine makes up 35% of DNA, then cytosine will be, आपको ये सॉल्व करना पड़ेगा. आपने देखा होगा चार्वर्स रूप में A इज इक्वल टू टी एंड G इज इक्वल टू सी. तो अगर एडिनिन 35% है तो आपका थाइमिन भी 35% होगा, तो टोटल हो गया आपका 70%. तो देन 100 माइनस 70 दैट इज़ 30. तो साइटोसिन हमारा क्या होगा 15%. तो साइटोसिन इज इक्वल टू ग्वानोसिन, तो गाइनोसिन भी क्या होगा हमारा 15%. बार इज़ अ हर्बिसाइड रेजिस्टेंस जीन. ये क्वेश्चन अभी रिसेंटली भी आपके ICR SRF PhD बायोटेक्नोलॉजी में 2025 में पूछा गया था. साइटोप्लाज्मिक चैनल्स कनेक्टिंग एडजेसेंट सेल्स आर कॉल्ड प्लाज्मोडेस्मेटा. Homologous pairs of chromosome become aligned to equatorial plate during metaphase 1 stage. To prevent the browning of culture medium because of the phenolics released some plants, polyvinyl pyrrolidone is used. PVP is used to prevent from the browning of the culture medium. A functionally comparable structure in plants to that of the yolk of a chicken egg which nourishes the developing embryo is endosperm. एंडोस्पर्म की तुलना की गई है आपके चिकन के योक से, एग योक से. Between two mitotic phases of cell cycle, the cell lies in G1 stage. In meiosis, as in mitosis, the chromosome material is replicated in S phase. Mutation that brings only one base pair change in DNA is known as point mutation. The diversity residing in DNA is because of the sequence of nucleotide pairs. In transduction, genetic material is transferred from one cell to the other through a phage. In transformation, no biological agent is involved for transfer of genetic material. Transcription factors control gene expression in eukaryotes. Tatum got Nobel Prize for his work on Neurospora crassa for one gene, one enzyme hypothesis. In conjugation, the free end of chromosome moves from HFR donor into F cell across mannitol was an intracellular bridge called pilus. The existence of pups in giant chromosome of the Brazilian goat support the concept that DNA unwinds for mRNA transcription. Agitation of the culture medium during cell suspension culture is done to maintain the uniform distribution of cells or cell clumps in the medium. Diazobenzyloxymethyl DBM paper is used in case of northern blotting. आई होप कि आप लोग साथ साथ में याद भी कर रहे होंगे और नोट्स भी बनाते जा रहे होंगे. A good molecular marker must be polymorphic, heritable, and codominant in nature. Physical map of a genome is based on recombination frequency and number of base pairs. During protoplast isolation, plant cells are treated with cellulase and pectinases. Shotgun approach is mainly used in genomic library development. Neck translation is useful for labeling of double-stranded DNA molecules. Acetyl coenzyme A carboxylase requires biotin as a cofactor. For mitochondrial structure and function, mitochondrial DNA specifies 10% protein. In transcription initiation process of eukaryotes, holoenzymes bind to the promoter. For both RNA synthesis and DNA replication, there is an absolute requirement of free 3' hydroxyl group. Replication of chloroplast DNA is controlled by nuclear DNA. The sigma factor of RNA polymerase in E. coli is involved in transcription initiations. The end process of transcription in prokaryotes is polycistrone in mRNA, and in eukaryotes it is monocistronic in RNA. The HSD system of DNA methylation in E. coli methylates adenine. The conversion of IMP to AMP requires GTP. Guanosine is a purine nucleotide. Jacob and Monod proposed operon concept in 1961. The sedimentary velocity of a protein in a centrifuge does not depend on the charge on the protein. Lactic acid is the major metabolic product of glycolysis in higher organisms. Roquefort cheese is ripened with the help of Penicillium roquefort. Bacteria are preferred over yeast for the production of single cell protein due to their fast growth. The function of the nucleus includes housing the hereditary information. Thiobacillus thiooxidans grows on pyrites. Baculoviruses are DNA viruses. In replication of E. coli DNA, the protein complex involved is replisomes. The number of consensus sequence in the promoter region of a gene in E. coli is generally 2. Up and down mutations occur in promoter region. In biotin labeling of probes, the detection method is streptavidin labeling. When PCR is used to generate single-stranded copies of DNA, this variation of PCR is known as asymmetric PCR. The evidence for semi-conservative replication mode of DNA was given by Michelson and Stahel in 1958. The enzymatic method of DNA sequencing was developed by Sanger and Coulson. Hybridoma technology is useful in the production of monoclonal antibodies. DNA reassociation kinetics provides information on the genome size. Degradation of xenobiotics occurs rapidly in well-aerated soils. Anther culture provides a method for the production of homozygous lines. Indole acetic acid is a naturally occurring oxime. An example of commercially important primary metabolites is alcohol. In PCR, double-stranded DNA is denatured by temperature. Nodulin genes are found in plants. Micropropagation involves initiation of aseptic culture shoot multiplication, rooting and transplanting. The shoot apical meristem is characterized by dome shape. The term soma clonal variation was coined by Larkin and Skovrov in 1981. Inclusions of osmoticum in both isolation and culture media prevents rupturing of protoplasts. Cryopreservation involves cutting, application of cryoprotectant and storage at minus 196 degrees centigrade. Explantation is a process of dissection and culture of small organs or tissue sections. The in vitro electron donor to PS1 is water. Acrid in orange is used for inducing frame shift mutations. The restriction enzyme useful to the molecular biologist belongs to type 2 class. Ferrous protein of nitrogenase is homomeric dimer. Leg hemoglobin is synthesized exclusively in nitrogen fixing nodules. The first product of purine nucleotide biosynthesis that contains a complete ring of hypoxanthine is IMP. So students, these all are the assessment of questions which are previously asked in ICR net in biotechnology examinations. So प्लीज़ लाइक एंड सब्सक्राइब माई चैनल इसी तरह के और शॉर्ट नोट्स प्रोवाइड करने के लिए और इसी तरह के और प्रीवियस ईयर क्वेश्चन पेपर को फिर से आपके सामने लाने के लिए आप जितना हो कमेंट करें। अगर आप और चाहते हैं कि और प्रीवियस ईयर के क्वेश्चंस पेपर में लाऊं 2001 से लेके 2025 तक, तो इस तरह से आप कमेंट करके मुझे ज़रूर बताएं।